Welcome to Season 5 of Public Health on Call, a podcast from the Johns Hopkins Bloomberg School of Public Health. I'm Joshua Sharfstein, Vice Dean for Public Health Practice and Community Engagement and a former health commissioner here in Baltimore, Maryland. Our goal with this podcast is to bring scientific evidence and experience to shed light on critical health issues. If you have questions or ideas for us, please send an email to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Today, our topic is convalescent plasma, the treatment that uses antibodies from people who have recovered from COVID-19 to help others. I speak to Dr. Arturo Casadevall from the Johns Hopkins Bloomberg School of Public Health, one of the leading experts and advocates on convalescent plasma. We discuss the latest evidence and what it means for patients. Let's listen. Dr. Casadevall, thank you so much for joining me in Public Health on Call to talk about convalescent plasma. How are you doing?
I'm okay, Josh. Thank you for having me.
So we heard about convalescent plasma early on in the pandemic on this podcast, and a lot has happened since then. So I want to go back a bit to the beginning and maybe just ask you to explain what convalescent plasma is.
It's a therapy that is made from people. When people recover from COVID, they have antibodies in their blood, and they can donate this plasma, and this plasma can then be given to other individuals for a treatment of the disease.
And I've been reading about high titer plasma versus low titer plasma. Can you explain what that is?
So what titer is a word for the amount of antibody. So when we recover, some of us make a lot of antibody and some of us makes less antibody. The best plasma comes from donors that have a lot of antibody, and we call that high titer plasma.
And you can figure out which plasma is high titers?
Exactly. You can take it into a laboratory. There are many tests, and you could just test it and see how much antibody it has.
The word antibody brings to mind monoclonal antibodies, which have sprung up and have been widely used and for many patients very effective. How does convalescent plasma compare to monoclonal antibodies in terms of what it is?
Antibodies are proteins of the immune system known as immunoglobulins. And they're normally made in infection for protection. You can also make them in the laboratory or in industry, and they're also known as monoclonal antibodies. But Josh, I want you to keep something in mind. Both plasma and monoclonal antibodies have the same active ingredient. Therefore, if one works, the other one should work. And that's something very important to discuss.
Great. Well, let's talk about the rationale for antibody therapy generally. When this all started, and there was, no vaccine on the immediate horizon. People were sort of trying certain types of treatments that were already available. Convalescent plasma popped up. What was your thinking in proposing this?
So antibody therapy in the form of convalescent plasma, also known as serum therapy in the past, goes back 130 years. We have a lot of experience with it. The ability to transfer antibodies can be used to treat disease. So whenever there is a pandemic or an epidemic, physicians often resort to it because it is available as soon as you have people who recovered. So it's relatively inexpensive, available, and there have been a lot of experience with it.
So take me all the way inside the body. What is happening in theory to help people who are getting antibody therapy, whether it is monoclonal antibody therapy or convalescent plasma?
So God forbid that you haven't been immunized and you get COVID. So what happens is you go through this initial viral phase when the virus replicates. The virus replicates in tissues, in the nose, sometimes in the lungs, and it takes a while for the immune system to begin to make antibodies. That often takes anywhere from 7 to 12 days. Now, if at that phase, you don't have a lot of protection, and the virus is replicating. Now, most people, the immune system catches up and checks the virus and they recover. But some don't. And those are the ones who then get massive inflammation and end up in the emergency room. So what you can do is you can give them antibody early in the process. That antibody neutralizes the virus. And then you stop the progression of the disease. And this is the premise behind monoclonal antibodies that prevent hospitalization, and it's the premise behind plasma, which is also to provide something that the body doesn't quite have.
In a way, it's giving the immune system a head start.
Exactly. It's giving them a few extra days to get going. And that could translate into avoiding hospitalization. And if you avoid hospitalization, you avoid death.
Now, that's the idea. And there are a lot of treatments out there in the world which have really good ideas behind them. And then they actually are tested, they don't pan out.
Right.
And so this has been, quite a period. A lot of studies have been done on convalescent plasma. And some of them have had positive results and some of them had had negative results. And so I wonder if you could kind of characterize the data that's emerged, thinking about also the progression of the patient from someone who, maybe was exposed all the way through to somebody who's already in the intensive care unit. You could go in reverse order, whatever you think best in terms of thinking about the state of the evidence.
Thank you, Josh. That's a big question, but let's just stop on how do doctors know what they know? How do you know that something actually works? So in medicine, medicine has made great progress, and we have layers of certainty. First, you could have experience. Then you could carry out what is known as an observational study. Do the patients do better if you give them plasma? And then you can create controls for them. And then the single most rigorous way of knowing something is what is known as a randomized controlled trial, often with a placebo control and ideally double-blinded to avoid bias. So to put everything in perspective, in the 130 years before COVID, we had one randomized controlled trial for plasma that was done in Argentina in the late 70s. Why? Because they're very hard to do in the middle of a pandemic. In the last 2 years, we have now completed about 25. So we have a lot of very high quality data. And when people look at this data, the important thing to know is that the positives and the negatives each teach you something. Because a negative trial shows you when it doesn't work. A positive trial shows you when it works. These are not, people get very worked up. Oh, the data is mixed, the data is conflicting. No, the data is the data. One just needs to look at it and process it. So we now have, almost 2 years into this, a wealth of information. And what I can say to you is, in one sentence, is that the wealth of information has confirmed what we already knew, that antibody therapies are most effective if used early and with sufficient antibody. And you can take all these 20 plus trials and put them in a row. The ones that treated late, you tend to see no efficacy, although you often find a hint here because they always include some population, some group that could have benefited. The ones that treated early tend to see big effects.
And why might that be? Before I start to ask you some specific questions about the studies, why would it be that this treatment would work earlier, but not later when people are really sick?
So, George, that has to do with the question you asked me before. The late in disease, people already have their own antibody. So giving them their antibody, you're giving them something on top of something that they have. They may get a little benefit of it, but that's not it. But the central problem is that COVID is a disease where what kills you is inflammation. Inflammation in the lung prevents gas exchange. You cannot expect antibody to reverse inflammation. All you can expect antibody to do is to neutralize the virus. And if you neutralize the virus, well, that prevents further inflammation. So the earlier you treat, the much more likely you are to be able to affect the process.
Now, the NIH did a study in 2020 and 2021 called the C3PO study and concluded that early use of convalescent plasma didn't work.
So when C3PO was first designed, we had great hopes for that study. Now, notice the word early. Early is relative, right? Because these people are already walking into the emergency room. So this study involved having people show up in the emergency room. Now they're sick enough to be in an emergency room, and then to be randomized to either receiving convalescent plasma or an infusion of some sort of vitamin water. And then the question was, if you release them, will they come back with COVID? That is, did you stop the progression of disease at that point? So it turns out that the way the study was designed, if you follow the rules of the study, you don't see a difference. In other words, those who got plasma or got vitamin water ended up doing roughly about the same. But if you look at what they actually did, is it was designed that people who showed up on that index visit counted if they got admitted. Now, it seems to me that if you go to an emergency room and you're sick enough to go to the emergency room and they put plasma into you and you get admitted, well, I don't think you can expect plasma to work right away. Imagine in the gurney. So if you now look at the same data and you remove those patients that were admitted on the index visit, C3PO has an efficacy of over 30%. That is, those who were discharged home were less likely to come back if they received plasma. And it all kinds of makes sense. The ones who get discharged home were probably less sick than the ones who get admitted.
In a way, your critique of that study is that they included in their primary analysis people who were actually pretty sick.
Exactly. And by the way, we have posted a critique of that study in our website, ccpp19.org. Those who are interested can go and look at it. But we in the field are considering that a positive study, even though the NIH is considering a negative study. The way we look at it is we look only at the data, period, and we interpret it.
Now, I'm sure you're aware the World Health Organization looked at this question of early use of convalescent plasma and decided not to recommend it on the basis of all the studies that were done in early December. And that was probably a big moment in this field. Why did they decide that, and how did you respond?
So I would say to you that we were very disappointed with the WHO position, and we don't agree with it. We don't agree with it. What they did is they took all the randomized control trials and they did a meta-analysis. And basically they ended up with concluding that there wasn't a lot of efficacy. Some of these randomized control trials were huge. If you do a meta-analysis, a huge trial that is negative totally wipes out the effect of the other trials that are positive.
Could you just explain what a meta-analysis is?
A meta-analysis is a way in which you have a lot of different observations or trials, and it's a mathematical way of combining them. And it's controversial. For example, a meta-analysis, if you were to take a bunch of randomized clinical trials and put them together, and then you ask the question, what is the likelihood that if you carried out a randomized clinical trial with that number of patients, that you will have a predictive value? It's only 50 or 60%. So as a tool, I don't necessarily think it has a lot of predictive value or rigor, but it is greatly used by committees, something that we disagree with because it doesn't have the nuance that is needed to try to understand this therapy.
And would it be fair to say that some of those studies may not have used the high titer plasma that you anticipated would have a bigger impact? So you're combining studies of different kinds to get sort of one overall answer.
Yeah, absolutely. Some of these studies use very late. Other studies use insufficient plasma. And I will tell you one thing, the WHO did not have access to the Hopkins data, which we're going to cover. But I will also say to you that we have reached out to them, and David Sullivan, the PI of the Hopkins study, has presented the data to them. And we are hopeful, since they are physician scientists and rigorous people, that they will issue new recommendations in the future.
So let's talk about the most recent study that you were involved with. Tell us about that project.
Okay, so to tell you about that, we have to go back to March, April of 2020. In those very dark days, a lot of people throughout the world set up randomized clinical trials for plasma, which is exactly what was important to do. Unfortunately, the great majority of them were set up in hospitalized patients. Hospitalized patients, by definition, are quite sick. Otherwise, you wouldn't admit them. Why do they do it in hospitalized patients? Because it's easier to do. You have the patient already in a room.
Right.
Imagine trying to do an outpatient trial.
Well, also at that time, testing wasn't so widely available, probably.
It wasn't available, and there was actually nothing else to do. So people were trying to do the best they could for patients. Here at Hopkins, David Sullivan, Shmuel Shoham, Kelly Giebel and others, set up to do a very different type of clinical trial, one involving outpatients. Very hard to do. Think about logistics. You got to take somebody who is infected and bring it to some place in which you're now going to give them a unit of plasma. There were no facilities for that. They had to set up tents outside of the hospital. And they set up a multi-center trial in many other parts of the United States. So it was a huge logistical effort. But it was the one trial who tried to test it in the conditions when it's supposed to work, which is early in the course of disease. And the trial took a while. It had its ups and downs because when the epidemic ebbed, there weren't sufficient patients. Then there came the vaccines and all that. But they were able to complete it by late October. It involves 1,000 people, or I believe over 1,100 in 2 arms. And the main result of the study is that those who received convalescent plasma as an outpatient had a 54 reduced likelihood of being hospitalized. In other words,
54% reduction.
Reduction in hospitalization. And it was double-blinded. Nobody knew anything. A data safety monitoring board followed things, and the control received plasma. That makes it extremely rigorous because received plasma prior to the pandemic, so it didn't have antibodies to the virus.
Let me just make sure I understand. By double-blind, you mean the doctors providing this to the patient did not know, and the patient did not know whether they were getting COVID-19 convalescent plasma.
Nobody knew anything.
And then the people who didn't get the COVID-19 convalescent plasma got plasma, but just from people before COVID.
Correct.
So there were no special antibodies for COVID in that.
Correct.
And when the dust settled, you looked at the results, there was a 54% reduction.
It was clear. I mean, it was highly statistically significant. And there is going to be subsequent analysis that makes that data even better if you look at it as a function of time. That is, if you got it in the first 3 days of symptoms, first 5 days of symptoms or so, which is exactly what we would expect. And is consistent with the monoclonal antibodies, which, by the way, were tested by the pharmaceutical industry in the most optimal conditions possible, which was the 3 to 5 days of symptoms.
So I heard you say you'll be presenting this to the World Health Organization.
It was done already.
Got it. So the World Health Organization may consider that.
Right. David Sullivan did the presentation to them. Yeah.
What about in the US, the FDA, and the potential to use this therapy now? Because particularly we've heard that monoclonal antibodies, some of them don't work for Omicron, and the ones that do work are in short supply. Is this something the FDA has considered for early in the disease?
Right. So within days of the Hopkins results being made public, the FDA moved to open plasma into the outpatient space. This is huge because it means that you can use plasma in the best conditions possible early in the course of disease. Now, here is another bit of good news. We don't need Omicron convalescent plasma. It turns out that people who had COVID and were subsequently vaccinated have such high titers of antibody that their plasma neutralizes Omicron. So we are now in a great position. We know how to use plasma. The FDA has opened the outpatient space, and we have a lot of donors who can give it. And as we lost 2 out of 3 monoclonal antibody therapies, and this provides a mechanism to hopefully help people from progression to disease so they don't have to be hospitalized.
And, just to circle on this issue of losing monoclonal antibody therapies, because they are such unique antibodies that the pharmaceutical industry makes, it is easier for a virus to mutate to evade them compared to the protection that you get from vaccination, which is broader, or in theory, the protection you would get from convalescent plasma, which has a variety of different antibodies.
That's right, Josh. When you get vaccinated or when you get convalescent plasma, you have thousands of different antibodies. When you get a monoclonal, you're getting 1 or 2, lots of it, but it's very easy for the virus to defeat it. And in fact, the SARS-CoV-2 virus has already defeated several of them. And we anticipate that new ones will be made. There is no question about that, but they will always be vulnerable to viral changes, whereas the plasma is the only therapy that keeps up with the variants.
So I want to pull back for the last question, Dr. Casadevall, which is, your perspective on what this whole experience that you've been involved in over almost 2 years now, how does it shape your view of science and how it advances? I mean, it has been ups and downs for this therapy. You've seen people respond to different reviews of data and make different conclusions on one side or the other. How has that affected your thinking about science?
So in some ways, it's made me very optimistic of what we can do. Things could be done very rapidly. People can respond and we can generate very high quality data very rapidly. On the other hand, it is highlighted to me that we have a tremendous amount of work to do in medicine on teaching physicians how to critically think about data, including, by the way, the recommendation committees, which provide recommendations, and I will argue to you, are often flawed because their way of analysis doesn't necessarily incorporate the way that you need a lot of these therapies. So what I'm hoping to, and as you know from other discussions we have had, I'm very interested in the problems of science and how scientists are trained. I think there has to be comparable efforts to get physicians to think critically about data and how, especially in a pandemic where the information is changing by the day.
Well, Dr. Casadevall, thank you for all of your work during the pandemic, and thanks for joining me on Public Health on Call.
Thank you, Josh. We had 3 minutes to spare. Anyway, thank you very much. I look forward to listening to it.
Public Health on Call is produced by Joshua Sharfstein, Lindsay Smith-Rogers, and Stephanie Desmond. Audio production by Niall Owen McCusker, Matthew Martin, Spencer Greer, and Holly Cardinale, with support from Chip Hickey. Distribution by Nick Moran. Production support from Catherine Ricardo. Social media support from Grace Holz Fernandez. Thank you for listening.